Hello everyone, so today we're going to be looking at antigens and the MHC. So we can see here our host factors also influence immunogenicity. So they can be repertoire related. So our repertoire is our total diversity in T and B cells in an individual. And this is our repertoire to be respond to an antigen. So you must have T and B cells that can recognize it. So this is all, as we discussed with B cells, the very many different mechanisms we have to have a huge range of receptors that can recognize many different antigens. And we also have MHC related, where we must have an MHC proteins that allow you to present the antigen. So you must be able to process the antigen, so the proteasomes and antigen transporters, which can make a difference, and we'll get into these later. So MHC genes are diverse in the population. So MHC genes between individuals are extremely diverse, and we'll talk about this later, but this is kind of the reason why there's so many issues with transplantation. So T and B cells. So B and T cells can recognize the same antigen, but in different ways. They see different types of epitopes. So as we discussed, epitopes are a binding site, or what we develop receptors for. So B cells see free antigen, usually surface epitopes, while T cells see peptide fragments, which are often internal epitopes. And these are complex with MHC protein, which is important to remember. So here we can see the differences between a T cell and a B cell. So, but there is exceptions to this T cell rule before I move on, and it's the gamma delta T cells. But here, again, we can see the antibody with our variable region, where the antibody's antigen binding site, and our constant region, which is effector function, which determines the class, which again, like for example, is IgM, IgD, and etc. Here we have our T cell receptor. As I'll learn about later, we got our alpha beta. Again, here's a variable region and our constant region. It's similar to the stalk of the Y, but you can see here there's membrane-bound components, and there's other differences in the formation, as we'll see later. So B cells' views of a lysozyme. So here this shows epitopes seen by 3 different antibodies on a lysozyme in this figure A. So in figure B, this is where a T cell views a lysozyme. The peptides in the lysozyme corresponding to 2 epitopes seen by the T cell receptors. So the blue is the epitope on the surface, and the orange is the epitope inside the fold of the protein. This epitope must be released by antigen-presenting cells, or processed by an antigen-presenting cell, and this peptide must be presented to T cells by APC using MHC proteins. So B cell versus T cell. So similar to the figure we showed earlier, we can see the differences between the antibody, or the B cell receptor when it's membrane-bound, and the T cell receptor. The B cell receptor, or antibody structure, versus the T cell receptor, we see the B cell receptor uses a membrane-bound antibody molecules to see antigens. The T cell receptor resembles a membrane-bound Fab fragment and of the antibody molecule, i.e. it has no Fc portion. The T cell receptor is a disulfide-linked heterodimer with each chain containing a constant and variable region. So you can see our variable and our constant regions. Similar in structure to the Fab fragment of the antibody, which is this fragment. The combined variable domains form the site for antigen recognition. So important, we'll get this later, but again, we have our Fab region in the antibody, antigen binding site, our VL, VH, CL, CH, and our FC region. And then in the T cell, we have our variable alpha, beta, C, and constant alpha, beta. So, structure of the T cell receptor. There are 2 chains which make up the T cell receptor. So, like we said, the alpha and beta chain, which are linked by disulfide bonds and a part of the immunoglobulin superfamily. It has a variable and constant regions, and the interaction between the variable regions of the alpha beta chains is where the antigen binding site occurs. So here the interaction. So the T cell receptor has a transmembrane domain, and the T cell receptor is not secreted, unlike the antibody. Each T cell has many copies of the same T cell receptor on its own cell surface. So here you can see the different regions. We have our carbohydrate, variable region, our constant region, the stalk segment, our transmembrane region, and our cytoplasmic tail. Again, merged through disulfide bonds. Some T cells have gamma delta chains. So here we have alpha beta. So there's 2 sets. There's our alpha beta T cells and our gamma delta T cells. Gamma delta T cells instead of the alpha beta, but these are usually seen in the mucus-associated lymphoid tissue and the epithelium, but we'll talk about them again later. So the binding site for antigens. There's a three-dimensional view of the T cell receptor showing the antigen binding CDR loops. So these are the complementary determining regions, which are highly variable CDR loops which recognize the peptide and MHC. Each alpha and each beta chain has 3 CDRs. It's important to remember. So again, CDR, complementary determining region. The T cell receptor gene rearrangements generate diversity is in the next lecture set. So that is, but yeah. So we have a second type of T cell receptor, the gamma delta. So T cell receptors of gamma delta T cells differ from the alpha beta. The overall structure is similar, but they use gamma delta chains, which have less diversity in structure. These gamma delta T cells are found in peripheral locations, which account for 5% of the total T cell population. They are in the epithelial locations, which accounts for 15%, and in mucosal location, it accounts for 40%. So they're mainly in mucosal tissues, like we said, the molt. They play a role in mucosal immunity and response to intracellular pathogens. The overall structure of the gamma T cell receptor, they're similar in structure, but are encoded by different sets of rearranging gene segments and have different functions. So essentially here, they're pretty similar, but you can just see it's formed by different chains. And the structure of both these types is similar, but the gamma delta T cell receptor has less diversity, so it's not as widespread and recognizes less pathogen, and does not require antigen to be presented to class 1 or class 2 MHC, which is important to remember. So T cells see peptides that have been processed and presented by MHC molecules, so they must use MHC. So here's an example. This is our peptide, our epitope, being presented by the MHC, then our T cell will come in and bind, as we can see here. T cells usually see interior epitopes released by antigen processing. These epitopes are seen by T cells and must be presented by MHC molecules, or major histocompatibility complex. This is important. This has an important impact on immunogenicity of antigens. T cells see only antigens that APCs can process and present. So many peptides in combination with MHC proteins. So here we can see the epitopes are recognized by the T cell receptors are often buried. So this is within the T cell receptor. The antigen must first be broken down into peptide fragments. This is done in the cell, and then the epitope peptide binds to a self molecule on an MHC molecule. So now it takes that peptide here, presents it to the surface. Then the T cell receptor binds to a complex of MHC molecule and epitope peptide. But for the T cell, this single binding between the epitope and the T cell is not enough. We need second signals. So the binding of a T cell receptor and to its cognate antigenic peptide requires simultaneous interaction of the peptide with both the T cell receptor on the T cell and the MHC on the antigen-presenting cell. So the portion of the antigen that contacts the T cell receptor is called the epitope, and the portion of the antigen that contacts the MHC is called the argitope. So here we can see this figure shows a side view of a T cell receptor interacting with an antigenic peptide presented by MHC molecules. And then figure B shows a B cell receptor antibody interacting with antigens. So we can see the differences. So some key points. Most antigens have more than one epitope. So more complex antigens have a wide variety of epitopes and have more, it means they're more immunogenic. As we discussed in other lectures, so certain complex proteins are the most immunogenic. The immune response to an antigen is equal to the response by varied epitopes. So B and T cells see different epitopes. So on the same pathogen, we could have T cells bind to one type of thing, while B cells affect another thing. B cells see free conformational or linear epitopes, which can be protein, polysaccharide, or lipid, where T cells see linear peptide epitopes produced by antigen processing, but only in combination with MHC proteins. But some T cells recognize lipid epitopes. So the MHC molecule. The MHC proteins are essential for antigen presentation to T cells. The MHC proteins must be both diverse and conserved. MHC proteins must be able to interact with many different antigenic peptides. MHC proteins must be able to interact with conserved molecules on T cells. These include the CD4 and CD8 T cells. So here we can get a picture of an MHC molecule. This is what is being presented in between. So again, MHC proteins are encoded by the major histocompatibility complex. So in humans, this is also seen as HLA. So you'll see this in the literature. They'll talk about HLA, and there's a bunch of different types. So this stands for human leukocyte antigens, or the HLA complex. In mice, this is referred to as the H2 complex, or histocompatibility 2 antigens. We'll focus on humans, but you will see this quite a bit. And a lot of the research came from mice, so that's also why it's relevant. Two major subgroups of the MHC proteins are used for antigen presentation. This includes our class 1 and class 2. And the key role of the MHC is antigen presentation, presenting the antigenic peptides to the T cells. So the two types of MHC molecules. We have the class 1 and class 2 MHCs, which have a similar 3D structure. So here we can see the differences. Clearly, first, the biggest difference is this beta-2 microglobulin, which we'll talk about later in the MHC class 1. So class 1s only have one polypeptide chain anchored in the cell membrane. There's 3 extracellular domains and associated protein that stabilizes the structure, which is the beta-2 microglobulin. So this is supporting stabilization. We have our class 2, which involves 2 polypeptide chains, and each is anchored to the cell membrane, and there are 2 extracellular domains in each chain. Major proteins involved in antigen processing and presentation are encoded with genes within the MHC. An example is the TAP, which is a transporter for antigen processing. We'll get to TAP later, but this is super important in MHC1 processing to bring the peptide to the surface, or pathogen, or epitope. So again, HLA2 and H2. A lot of these textbooks, figures, they will compare the human with the mouse, and you can see a lot of the terminology is different. DP, DOA, DM compared to the mouse were different, but we'll talk about these later. So again, we'll go over some terminology and organization in the class 1 MHC. So the class 1 MHC proteins are constitutively expressed on almost all cells of the body, and the MHC1 present peptides to CD8 T cells. So important to remember, MHC1, essentially just memorize this. You'll probably learn this in physiology as well. This is a more basic thing they teach in pretty much any immunology context. And 2 is the CD4. I guess an easy way for me to remember this is 4 divided by 2, MHC2. And then the other one is CD8. Maybe that helps. So in humans, there are 3 types of class 1 MHC molecules. There's type A, B, and C, usually encoded and referred to as HLA-A, B, and C genes. In mice, there are 3 types as well, K, D, and L, and this is H2K, H2D, H2L. There's codominant expression, so all 3 types are usually expressed on each cell. So each cell will have multiple MHC1 molecules. It's not just the one type, and alleles from both parents are expressed. So virtually all cells in the body express MHC class 1 proteins. Most mice and humans have 3, but different genes code for them. So again, here we see the different codes. So again, we can see up here, human HLA gene, we can see our different types. So again, we have class 1, class B, class C molecules are highly polymorphic and are used to present human peptides. So here's a figure displaying all the human versions. So A, B, C, E, F, G for human HLA. So class 1s again present the CD8 T cells. Class 2 include DP, DQ, and DR molecules, which are highly polymorphic and are used and present peptides as CD4 T cells. So again, remember, they all contain that beta-2 microglobulin, where the MHC includes the alpha beta. So now we'll look at the class 2. So class 2 MHC proteins are expressed mainly by professional antigen cells. So there are APCs, so dendritic cells, macrophages, and some B cells. These present peptides to CD4 T positive helper cells. So in humans, there's DP, DQ, and DR, and these are coded by HLA, DP, DQ, and DR. In mice, there's A and E. So a difference here, unlike MHC1, is that there's 3 in humans, 2 in mice. There's co-dominant expression, so all types are usually expressed by each APC. So each APC will present, our DQ, DP, and DR. And we note that antigen-presenting cells express class 1 also. So antigen-presenting cells are not only just MHC2, they have both. So the 2 classes of MHC proteins are expressed differentially on cells. So hepatocytes located in the liver express low levels of class 1 MHC. This kind of explains why their liver transplants are quite successful in comparison to other ones. We also have our microglia, which are differentiated macrophages, and these are in the brain, and these express class 2 MHC. T cells in humans can express class 2 MHC when activated. So class 1 MHC is expressed on all nucleated cells, and the highest levels are seen in hematopoietic stem cells, or cells. Class 2 MHC is expressed by antigen-presenting cells, and also on thymic epithelial cells. So here's a really good summary figure. So the darker the color and the more positive signs, the more expression. So we can see our MHC class 2 is highly expressed on our T cells, B cells, macrophages, dendritic cells, but also we have lots of MHC2 on their antigen-presenting cells, which are these 3 right here, and also in the epithelium, which we talked about here, the thymic epithelial cells. Our neutrophils have high expression of MHC class 1, so these phagocytose pathogens and can present it with MHC to the T cells, initiating an adaptive immune response. They're also seen on hepatocytes, the kidney, and the brain. And again, one interesting thing here is our non-nucleated cells, like the red blood cells, don't have either. So here again is a quick structure of the MHC class 1 and class 2. So CD4 means this will be MHC 2, CD8, 1. So here we see our beta 2 microglobulin. We'll talk about this soon, but here are co-stimulatory. This is the CD8 binding, but we'll talk about later that we do need some other signals to complete the process. So here's our T-cell receptor. T-cell receptor, class 2. We can see the difference in the beta 2 microglobulin. And we have CD4, which is a different shape. So the class 1 is a transmembrane. Class 1 MHC is a transmembrane glycoproteins with 1 long chain and 3 extracellular domains, alpha 1, alpha 2, and alpha 3. So here we can see 1, 2, 3. And then we have that beta 2 microglobulin again, which is very important in stabilizing the class 1 molecule. It has a peptide binding group. And then here's the more detailed figure. You see the structure of the proteins. We have a schematic diagram. The class 1 MHC proteins have a peptide binding groove, which is formed where the alpha 1 and alpha 2 domains interact, which is right here. So this peptide binding groove is formed by the interaction of alpha 1 and alpha 2 domains. It involves the walls of the alpha helix and the floors of the beta sheet. So allele-specific pockets bind peptides. So MHC1, just to point this out again, is closed groove, which we'll see this is important in comparison to MHC2. So MHC1 closed groove. So this binds short peptides, only 8 to 10 amino acids long, and the ends of the peptide bind to the class 1 MHC molecules and is pinned down by the class 1 MHC. So again, we have a structure, alpha 1, alpha 2, alpha 3, and beta 2 microglobulin. We have the small peptide binding groove that can look at short peptides. So more about class 1. The class 1 proteins are linked to a beta 2 microglobulin molecule, which resembles an immunoglobulin domain. The antigen binding groove in the MHC1 molecule protein is formed by 2 walls of a long alpha helices resting on the floor of the 8-stranded beta sheet, like we said. The peptide presented by the class 1 MHC proteins fit into the groove between alpha helices, so again, closed groove. The peptide backbone is bound at both ends by highly conserved regions of MHC protein. Some peptide side chains extend downwards into specific binding pockets in the groove, while other peptide side chains project upwards, where they can be recognized by T cells. So here again, peptide binding groove. We have our short peptide in the beta sheet. So again, it binds to short peptides, only 8 to 10 amino acids long, bound at their ends. So class 1 MHC is used for antigen presentation to CD8 positive T cells, as we said, and these are the CD cytotoxic T cells used for killing. The CD8 on the CD8 positive T cells binds to the invariant portion of the class 1 MHC, or the alpha 3 domain. So here we can see CD8 binds to alpha 3. The CD8 positive T cells must interact with the class 1 MHC. They are a terminology commonly used as class 1 restricted, so they will not work without MHC class 1 activation as well. So again, what type of antigen do they present? So we know how they function, so I think it's simple enough to say that they present endogenous antigens. So these are antigens that are synthesized in the cell, including intracellular pathogens and viruses. So again, as we talked about in the neutrophil, that neutrophil pathogen goes in, gets phagocytosed, MHC1 is presented, presents that antigen. So now it came from endogenously, and then our T cells come and bind, and then it started to adapt immunity. So here we can see, now we're going to be looking at class 2. So the class 2 MHC has a transmembrane glycoprotein, which includes 2 chains. The alpha and beta. Sorry, you can hear the cat. Each chain has 2 extracellular domains, the alpha 1, alpha 2, beta 2, beta 1. So here we can see that. The class 2 MHC proteins have a peptide binding groove, which is formed by the interaction of alpha 1 and beta 1 domains. So here's the schematic diagram down here if you want to take a closer look. So the peptide binding groove in class 2 is formed by the interactions of the alpha 1 and beta 1 domains. The walls are lined with alpha helices, and the floor is made up of beta sheets, similar to the class 1. It is also allele-specific pockets which bind peptides, and in comparison to MHC1, which had a closed groove, MHC2 has an open groove. So this means that it can essentially bind longer peptides, 12 to 17 amino acids long. So before I get to this, so we can see here the longer peptide binding groove that allows it to move, to bind longer MHC peptides. So what they kind of do is they compare MHC1 and MHC2 as a hot dog in a bun. So I'm going to do a crappy drawing, but so this is MHC molecule, so the hot dog in is within the bun. But in MHC2 with the open groove, the hot dog basically extends beyond the bun, but that's just a good way to remember it. As you'll see here, the peptides are longer and they can extend beyond the molecule. So MHC class 2 proteins have overall similar structure to MHC class 1 proteins, as we talked about with the alpha 1 helices and the beta plated sheets. Although their subunit structure is distinct, the MHC class 2 proteins are comprised of 2 subunits that contribute to the structure of the head domain containing the antigen binding groove. As a rule, MHC class 2 proteins bind longer peptides, as we said, than the MHC class 1 proteins. The different shape of the antigen binding groove in class 2 proteins allows the ends of the peptide to stick out, kind of like in our extremely poorly drawn hot dog example. The MHC class 2 proteins display peptides on the surfaces of specialized antigen-presenting cells and activate helper T cells. So again, MHC2 to CD4 positive T cells. So now we're going to kind of compare them. So again, here's kind of the hot dog analogy again. So in MHC class 1 molecules, in A and C, both ends of the peptide are tightly bound either end of the cleft. In MHC class 2, the molecules here in B and D, the ends of the peptide are not tightly bound and the peptide extends beyond the cleft. So the peptides are tightly bound in an elongated conformation in the peptide binding groove. The upper surface of the peptide MHC complex is recognized by T cells. The T cell receptor sees this combination of the residues of the MHC molecule and the peptide. So again, they're MHC restricted, so we need both the signals. So here's an example of CD4 binding to a beta 2 domain of the MHC. So again, CD8 binded to alpha 3, where in CD4 we bind to beta 2. The class 2 MHC is used for antigen presentation for CD4 positive T cells, like we said, or T helper cells. These CD4 positive T cells must interact with antigen and the class 2 MHC on the antigen presenting cells. CD4 binds to the invariant portion of the class 2 MHC, which is the beta 2 domain. So MHC1 was endogenous, and in MHC class 2 is exogenous. So these are synthesized outside the cell and taken in by phagocytosis or endocytosis. So extracellular pathogens and components include many bacteria. So MHC class 2 molecules are on professional antigenic cells, antigen-presenting cells, and interact with CD4 and T helper cells. Where MHC class 1 molecules are on pretty much all our cells and interact with CD8 or cytotoxic T cells. So again, here's a good figure comparison here. Here's our CD4 and our MHC class 2 binding to the beta 2 region, where our CD8 and our MHC class 1 binding to the alpha 3 with the beta 2 microglobulin stabilizing. So again, we've brought up this topic with MHC restriction, meaning that they need the MHC. So this figure, we can follow along here. The T cell receptor recognizes, so here's a recognition, recognize a complex of antigenic peptide in a self-MHC molecule. So then we have MHC restriction, where a T cell is specific for peptide X. So for example, here's our peptide X. And MHC allele, MHC A, there we can see that, will not recognize the complex of peptide X with a different MHC allele, MHC B, on the panel, which is the central panel here, or the complex of peptide Y with the MHC A. So we need the exact fit between both the MHC and the complex. So not just one or the other, as we can see here. So we need these MHC proteins to be able to bind to a wide array of peptides. So MHC proteins bind to a much wider range of peptides relative to the B cell and T cell receptor. They manage this because MHC proteins are flexible rather than rigid. Conformational changes occur to accommodate peptide binding. So also, only a few residues of antigen peptides are in close contact with the MHC peptide binding groove, and these are anchor residues. These anchor residues are similar between different peptides binding a given MHC protein, but the rest can be very different. So here's just a bunch of possibilities of where it can bind. So there's a lot of flexibility in where it can actually bind to pathogens or antigens. Any single MHC protein cannot bind to every peptide you might encounter, which seems kind of obvious. If you can't bind with it, your MHC, you can't present it to your T cells, so you'll never get a response. We deal with this through polymorphism and polyginy. So polymorphism is when we have many different alleles for each MHC protein. This allows for variation in MHC structure at the peptide binding groove. Polyginy, which is the presence of several different related genes with similar function, allows each individual to make a range of different MHC peptides. So here's a picture depicting these different phenomena. But we usually have both of them at the same time, so this allows for even greater diversity. Again, the result is diversity of MHC within individuals and populations. So MHC polymorphism. There are multiple alleles of MHC genes at each gene locus, so the MHC is highly polymorphic. The expression of MHC genes is co-dominant from both parents, and express your gene products from both parents, as I just said. The result is a high degree of MHC polymorphism in populations and individuals. So even family members can have extremely different MHCs and even have difficulties with transplantation within families. So again, we have our class 1 MHCs and our class 2. And remember, we had our DQ, DR, and DP, and then these are all expressed in multiple variations. So you can see, even though they have the same things, we can see they're expressed. So here's our C. As we said, we also have our HLA. So remember, these all start with HLA, and then our class 2s are in the D, and then class 1s use C, L, and C, A, and B. So here's a good illustration here, kind of confusing, but we'll go through it. So MHC co-expression is co-dominant. We have 3 different class 1 MHC proteins, again, A, B, and C. Each cell expresses both parental genes. So that means we have 6 total. So the A, B, and C from our maternal, and the A, B, and C from our paternal. So this allows for many different combinations. So the class 2 proteins, which are DP, DQ, and DR, each have 2 chains, alpha, beta. Both parental contributions are also expressed. So we have DP, DQ, and DR from maternal, DP, DQ, and DR from paternal. So again, this allows for so many combinations. So this can be our maternal, paternal, and again, same thing. So there's a lot of options available. And additional combinations can occur if the alpha-beta chains from each parent are possible. So we can have our alpha maternal and alpha paternal, or we can have alpha M, beta M, and so forth and so forth. And then this can be P, this can be M. So there's numerous options if we could do the math. So this MHC polymorphism results in variation in the MHC protein binding groups, which we discussed earlier. MHC protein polymorphism of the MHC is the greatest in the regions of the MHC protein that interact with the antigenic peptide. So a lot of the other structure won't be as variable, but this is where the most variation occurs, where it needs to bind to different antigens. The red here, these red little squares, indicates the amino acids oriented toward the peptide binding groove. These are the most variable areas between the different MHC alleles. So again, here are those red areas, highly variable. The variation of the MHC protein structure contributes to a wide range of peptide binding. This variation is concentrated in sites that bind peptide in the T cell receptor. The variable sites, the red dots, line the peptide binding groove and influence peptide binding and interaction with the T cell receptor. So in humans, the DR alpha chain is monomorphic, but the HLA-DP and DQ alpha chains are variable. So here's our class 2, and here's our class 1. So allelic variation occurs at specific sites within MHC molecules. Variability plots, so here we can see our variability plots between the different regions, our alpha 1, alpha 2, and A3, and our beta 1 and beta 2. So here we can see the red variability. So you can see certain areas or residues are more polymorphic than the others. So variability plots of amino acid sequence show the polymorphism localizes to the domains forming the peptide binding clefts. So the alpha 1 and alpha 2 domains of the class 1 MHC and the alpha 1 and beta 1 of class 2 domains. However, HLA-DR alpha chains lack variation. In the figure above, only the beta chains show polymorphism because they're shown in an HLA-DR molecule. So that's just a little nitpicky, but you can see here where the variation occurs, and as you said, alpha 1, alpha 2, this is where we have that formation. And we can remember alpha binds to the CD8, alpha 3. So this polymorphism also influences the response to the antigen. So the selective binding of processed peptides by different MHC class 2 molecule alleles we can see up here. Protein antigen, processed peptides, and expression here. So you must have MHC proteins that will bind to a peptide in order to present your T cells. Like we said, they're MHC restricted. This can lead to variation in responses. Different peptides/epitopes are immunodominant in different individuals. Is there an advantage to this diversity? It likely is at a population level. This diversity at MHC alleles helps ensure a wide range of peptide presentation. So what is the point of this polymorphism? So individual to the individual is that each person expresses different types of class 1 and class 2 molecules, which results in an increased potential for antigen presentation, which is of course good for us to fight any array of pathogens we may encounter. Advantages to the population include that an antigen that cannot be presented by one person's MHC will likely be presented by another person. This decreases the chances of a given antigen that cannot be presented by anyone in the population. There are limits to the number of peptides any given MHC protein can present, i.e. some antigens cannot be presented by some MHC proteins. Polymorphisms provide a strategy to deal with this problem. So if we had a virus that somehow was not bound to any human's MHC, we pretty much would all get infected by the virus, and there's nothing you can do. But even if we have a terrible virus or some terrible virus that wipes people out, there's still going to be people that have MHCs just through natural diversity that will recognize it, and they would be immune and have a response. So, for example, there's some cases of people who've caught HIV or AIDS that actually are somewhat resistant, basically just to the luck of the draw, and these people are used a lot for studies to think of ways to actually fight AIDS. So here's another example. So this occurs in animals in captivity quite a bit. So examples, cheetahs, whales, in wildlife in isolated locations. So certain moose populations. So here we can see what basically happens is that there's inbreeding between these animals and they lose their MHC diversity. since they're passing down the same MHCs over and over again, same genetics. So what results is that these viruses either mutate or certain diseases, basically they lack an MHC4 and it can wipe out essentially a population. So this inbred could cause a genetic basis for species vulnerability. So if a certain pathogens get to these species, they might not be able to deal with it. So this loss of MHC diversity causes increased vulnerability of population to infectious diseases. Interestingly, some antigens are not presented by MHCs. This includes lipids. Lipids can be presented by antigens by MHC-like proteins. This includes CD1. So CD1 is a non-polymorphic molecule expressed on antigen-presenting cells. The structure is very close to MHC1, as you can see here. Still has that beta-2 microglobulin, but slightly different structure here. It is also stabilized by beta-2 microglobulin, as we said. It has a deep hydrophobic binding groove, which binds lipids and glycolipids. It's important because certain bacteria, like Mycobacterium, infections have a high lipid content in their cell walls. So we still need a way to fight these infections. And these can interact with both alpha, beta, and gamma delta. So another view of CD1 here. CD1 presenting a synthetic lipid, so this is kind of experiment here. The CD1 family of MHC class 1 molecules is encoded outside the MHC and presents microbial lipids to the CD1 restricted T cells. The structure of CD1 orients the bound glycolipids so that carbohydrate groups protrude outward from the binding groove, and these are seen by the T cell receptors. You can see it protruding. There's also something called superantigens. So superantigens act as polyclonal activators of T cells. Examples of superantigens include Staphylococcus aureus, toxic shock syndrome toxin, or TSS-T1. So superantigens work because they bridge T cells to antigen-presenting cells by attaching to the MHC class 2 on antigen-presenting cells and to conserve portions on the T cell receptor. And these are on certain T cell beta receptor chains. You can see here binding to the conserved portion of the T cell receptor variable beta chain away from the CDRs and to the outer face of MHC. See that here? Bacterial superantigen and viral superantigen. You can see there's a difference in binding. We'll see. So structure and action. So example here is Staphylococcus aureus, Staphylococcal enterotoxin, or SEB, and SEC. In turquoise, they bind into the class 2 MHC alpha chain, seen in yellow here, and directly to the T cell variable beta chain in gray. The result is that they have stimulation of many CD8 T cells and cause excessive cytokine production and symptoms of toxic shock, including high fever, drop in blood pressure. So essentially, these things bind to so many T cells and so strongly that they pretty much cause intense activation of our response. So a massive amount of T cells get activated, and we basically have a cytokine storm, similar to what was seen and talked about in COVID. So MHC polymorphism is important to allow us to present diverse range of antigenic peptides. MHC structure allows for recognition of diverse range of peptides, but still allows interaction with conserved molecules on T cells. These are CD8 and CD4. Exceptions to the standard MHC presentation allow for unusual antigens to be recognized. As we mentioned, there are CD1 presentation of lipids and glycolipids involved in certain bacteria. And we also have our superantigens, which manipulate this process by bridging the class 2 MHC to large numbers of Th2 cells, or Th helper cells. MHC proteins present antigen processed through different routes, and processing determines interactions, and we'll get to this next time. So that's the end of the lecture. Hope you guys enjoyed it. MHCs are pretty much vital to our entire adaptive immune response, and they kind of get breezed over in a lot of lectures because people just tend to more focus on the T and B cells, which are obviously super important, but these are key to the process. And we'll look at their different interactions later and how we need co-stimulatory interactions. So it's not just the MHC only, but yeah, we'll get to that later. See ya. Take care.